we started zooming in on one of the main functions of proteins as enzymes. And we looked at the classification and naming of enzymes. And we also looked at enzyme specification, looking at the different forms of specification of enzymes. And with that specification today, we will continue to look at enzymes, and hopefully by the end of this meeting, we should be done with this portion of enzymes in the curriculum, so that the next time we meet, we'll continue to look at another function of proteins in the transport of oxygen. That is going to be looking at the molecule hemoglobin. So where we ended particularly on enzyme specificity, how enzyme specificity is explored in medical practice. Just one example. And this is particularly on the enzyme acetylcholine esterase. Acetylcholine esterase. And acetylcholine esterase is involved in the hydrolysis of acetyl. So that when acetylcholine is attached to muscle cells, they give the signal to the muscle to contract. And then over time, acetylcholine esterase, carries out, catalyzes the hydrolysis of acetylcholine. So acetylcholine breaks down into acetic acid and choline. That way, acetylcholine eventually leaves the receptors of the muscle cells, so that the muscle cell is ready to receive another signal from another molecule of acetylcholine to contract again. So there is that contraction, relaxation process, which is being induced by acetylcholine as the signaling molecule. Now, we have a compound which is succinylcholine, which is, you can look at it as a dimer of acetylcholine, because if you look at the structure of acetylcholine and you come to look at the structure of succinylcholine, you will see that succinylcholine is a combination of two molecules of acetylcholine. So two molecules of acetylcholine bind together to give you the structure of succinylcholine. So that is like a dimer of acetylcholine. Now, the enzyme acetylcholine esterase is able to act on succinylcholine just like it acts on acetylcholine. However, as you can see, succinylcholine is a bigger molecule than acetylcholine. And so it takes a much longer time for acetylcholine esterase to break down succinylcholine into acetic acid and choline so that it leaves the muscle cells to be ready for another signal for it to contract. So in that manner, succinylcholine is employed as a muscle relaxant used in minor surgeries in medical practice because succinylcholine, when is injected into the muscle tissue, it goes to bind to the receptors of the muscle cells, the same receptors that acetylcholine will bind to. Because it takes much longer time for acetylcholine esterase to hydrolyze succinylcholine, the muscles. One moment, I can hear somebody's voice. So just a moment, let me go check my settings and make sure that everybody's mic is off. Now. Okay, thanks. That is just some technical error because all the mics are off. Okay, so as I was saying, it takes much longer time for succinylcholine to be hydrolyzed into acetic acid and choline by acetylcholine esterase. So in that manner, minor surgeries that are being conducted when succinylcholine is injected into the muscles will be relaxed for the period of the surgery before by the time all the succinylcholine is broken down into acetic acid and choline and leaving the muscle cells to receive signal from acetylcholine to start contracting and relaxation again, the surgery would have been done. So that is a practical application of the knowledge on enzymes, their specificity, their activity in the body. So this is just one of them. As we go ahead, we'll see a couple of examples pertaining to the application of enzymes in other ways. Right. We continue to look at coenzymes and cofactors. In our last lecture, I briefly mentioned cofactors and coenzymes, and I made a point that even though these two terms are used interchangeably, in some biochemistry textbooks, you see a distinction between coenzymes and cofactors, and that is actually how it should be. Coenzymes are organic substances, whilst cofactors are inorganic, usually metals. So these are metal ions which are needed. So that's the difference. Cofactors are some metal ions, whilst coenzymes are some organic compounds. And they are the non-protein part of enzyme, which is needed in order to make an enzyme active. So we have the protein part of the enzyme we refer to as an apoenzyme. I think we looked at it when we were looking at the terminologies right after the introduction to enzymes. We looked at terminologies so that we'll be able to understand as the topic continues. So sometimes you see the apoenzyme is also called apoprotein. So that is the protein part of the enzyme. Now, usually you have that apoenzyme being inactive. It's being inactive, and you will need either a cofactor or a coenzyme, which is a non-protein. So sometimes it's being referred to as a prosthetic part of the enzyme. So that's coenzyme, cofactor, a non-protein portion is an activator. So when it attaches to the protein part, that apoenzyme, it forms a hollow enzyme that is an active enzyme. So the hollow enzyme becomes active. So you see the coenzyme or the cofactor being depicted here, which binds to the active center of the enzyme. Here you see there's a mistake in the diagram, such that you see that there's a coenzyme here. This coenzyme being written here, but here becoming red is shown as a substrate, but actually that shouldn't be a substrate. It's a coenzyme or a cofactor that binds to the enzyme to make it active before a substrate can bind in there, and the reaction can be catalyzed by the enzyme. So this is how coenzymes and cofactors act. They bind to the protein part of the enzyme, and they act as activators. So they make the enzyme active in order for the enzyme to carry out its functions. So if you look at it this way, that we saw different levels of the structure of proteins. We saw the primary structure of proteins, secondary structure, tertiary structure, and the quaternary structure. And you realize for primary structure, it is just the sequence of amino acids, which is in a peptide chain. And the secondary structure, you saw it's either some alpha helix or beta sheets. But it is actually from the tertiary structure and the quaternary structure that you begin to see functioning of proteins, because this is where you see proteins folding to have some specific structure, specific form that will begin to be active in performing their specific functions. And coenzymes and cofactors are involved in these two structures, or say the tertiary and the secondary structure, so that if those metals, the metal ions, which are the cofactors, or those organic compounds, which are coenzymes, around which you find the protein folding, having a specific form to have a function, if those are not there, then it means the protein loses its structure, its functional structure. So we saw some example of, say, denaturing. You apply heat, and then the protein loses its structure. And once the protein loses its structure, it will lose its function. All right. So we continue to look at some examples of cofactors. So these specifically are some metal ions, say some copper ions. We see ferric ion, Fe2+, ferrous ion, Fe3+. These two ions, we see all the ions over here, the copper ions, the ferric ions, potassium, magnesium, manganese ions, some molybdenum atom, nickel ions, selenium atom, zinc ions. All of these serve as cofactors to some specific enzymes, which are listed here. So really, it's not a thing to cramp into your head that to know that copper ions serve as cofactors to cytochrome oxidases, or and ferric ions serve as cofactors also to cytochrome oxidase. So you can see there is some overlapping. You see that copper ions serve as some cofactors to cytochrome oxidases. The same cytochrome oxidases, some of them you have ferrous and ferric ions serving as cofactors. So it's not a thing to actually be memorizing. These things are there, like you can easily refer to them if you need to work with any of these enzymes or metal ions. Experimentally, you are doing something in the lab that you want to do some analysis that it requires that you know which metals are required as cofactors or coenzymes. You can easily refer to literature and see them. So don't go wanting to memorize these things in any way. When you work with them for some time, some of them stick. And so that's just for you to know that these metal ions act as cofactors to enzymes, meaning that they are required to activate these enzymes to carry out their functions. Or in other words, they are required for these enzymes to have their functional structures. So they're supposed to, an enzyme will be active only when it has its functional structure. Once that functional structure is broken, which we refer to as denaturing, then the enzyme becomes inactive. Right. So, but one thing you can pay attention to, after all, we did enzyme naming and classification. So you can, when you look at the names, you can remind yourself of what type of reactions these enzymes will be carrying out just by looking at the name. Like, for instance, if you look at cytochrome oxidases, then you can say this will be involved in some oxidation and reduction reactions. And then you can also do that classification and say cytochrome oxidase is an oxidoreductase, because we did classification. So that way you can have a better understanding of what we learned earlier about enzyme naming, like pyruvate kinase, for instance. I mentioned that kinases are involved in the transfer of phosphate groups from one molecule to the other. Or in other words, they are involved in phosphorylation reactions, which you see often in the activation of molecules to go into biochemical reactions. And you can also tell that that enzyme will be transferring phosphate group onto, say, pyruvate, for instance. So you can do this this type of revision just by looking at the names. Hexokinase, you have seen transfer of phosphate groups onto hexoses, 6 carbon sugars, and so on and so forth. So you can do that. Then we see coenzymes. Coenzymes. So what we just looked at in the previous slide was cofactors. Those were metal ions. Now we are looking at coenzymes. So the coenzymes, I mentioned that they are some organic compounds. So you see the difference, that cofactors are metal ions, but coenzymes as some organic substances. Now we see these coenzymes that are listed here: biocytin, coenzyme A, all the way to thymine pyrophosphate. And you realize that on the last end of this table, the right-hand side, we see what is referred to as a dietary precursor. So that we can actually say that what we see at the right-hand side is an inactive form of the coenzyme. So for instance, if you see biocytin, and we see on the right-hand side, which is biotin, biotin is an inactive form of biocytin. Then another thing that you realize is that all these precursors, these inactive forms of the coenzymes, are some form of vitamin B. So all the vitamin B's, they act as coenzymes to different enzymes. So, for instance, we see biotin. Biotin, I don't remember so well, but I think biotin is vitamin B3. Pantothenic acid, I think, is vitamin B5. Then you see vitamin B12. You see riboflavin, vitamin B2. We see a coenzyme lipoid, that's to say it doesn't require any dietary. There's no dietary requirement there. So the lipoid is some lipid on its own. We have nicotinic acid. I think that is vitamin B7. You see pyridoxine, vitamin B6, folate. I think that is vitamin B9. And you see thymine, vitamin B1. So all of them, you realize that they are, all some form of vitamin B. So that should give you the understanding of why many a time when drugs are prescribed for patients, they are also giving, say, B complex, say vitamin B complex, which comprises of, it's a combination of all these vitamin Bs together. Because a person that is not well, a patient, must have some system dysfunction, and so that if a drug, a chemical, which is given to the person to take into the body, there should be some boosting of the enzymes that are involved in the metabolism of the drugs that are given to such individuals. And it is these vitamin Bs that act as activators of the enzymes. So that if the person is given a vitamin B complex, that with having adequate amounts of all these vitamin Bs, then of course they will serve as some form of activation of the enzymes that are involved in the metabolism of the drugs that are given to these patients. So we see each of these coenzymes, carry out some specific reactions. They are involved in some specific reactions, which are, of course, enzymatic reactions. So, for instance, biocytin is involved in the transfer of carboxylic acid groups. So it's written as CO2, but that is actually carboxylic acid, COOH group that in aqueous environments exist as an ion, the carboxylic ion, which is C double bond O minus. That's why you see it being written as CO2. Coenzyme A is also involved in transfer of acyl groups. We see deoxy adenosylcobalamin. So even the name, this name for coenzyme B12 cobalamin tells you that there is even in this coenzyme there is a metal ion, and that is cobalt, and this is involved in the transfer of hydrogen atoms, or say protons, and some alkyl groups like methyl group, ethyl group, propyl group, etc. So somehow you can see, once you see like there's transfer of some hydrogen atoms there or protons, obviously that is some form of reduction reaction. So means that this coenzyme is involved with some oxidoreductases. We see also flavin adenine dinucleotide, which you usually see in biochemical reactions written as FAD. Sometimes in its hydride form, you see FADH being written, also involved in the transfer of electrons. That tells you that it is involved in oxidation reduction reactions. Lipoate also, transfer of electrons and acyl groups. Nicotinamide adenine dinucleotide. That too you will see in biochemical reactions. You see being written in its abbreviation NAD, and also in its hydride form you see it NADH, also involved in oxidation reduction reactions. Pyridoxal phosphate involved in the transfer of amino groups. So when you go forward and you're looking at metabolism of biomolecules, you look at metabolism of proteins, there's a process called transamination. Sometimes it's together with, it's referred to as trans-deamination, because you have, see the amino acids have the amine group and the carboxylic acid group, and the removal of the amine group from these amino acids, you see this pyridoxal phosphate acting in that process of the transfer. You see tetrahydrofolate. See, tetrahydrofolate is what we see being prescribed as folic acid. So you see, folic acid is involved in the development of the fetus. So you see that a lot of times in pregnancy, folic acid is prescribed for some pregnant women to be taking because it's very much involved in the development of neural tubes. That is the brain of a developing fetus. So you see also is some form of vitamin B. I was saying vitamin B9, which is involved in that process, and it's involved in the transfer of some one carbon groups. So any group with just one carbon in it, and carbon is involved in cell proliferations. It's involved. I mean, carbon forms the skeletal structure of many organic compounds. Take carbohydrates, take proteins, take lipids. You see it's a lot of carbon skeleton upon which other atoms are bonded onto. Then we see thymine pyrophosphate also involved in the transfer of this functional group, the aldehyde groups. So when it comes to metabolism of carbohydrates, obviously you find it there. So for instance, if you take glucose molecules, if you look at the structure of glucose, say the linear structure of glucose, you see that that is an aldohexose, meaning it contains this functional group, the aldehyde group. And because it's made up of 6 carbons, it contains 6 carbons, we refer to, say, glucose as an aldohexose, for instance. So you find this aldehyde group in many biomolecules, so that in their metabolism, the transfer of these aldehyde groups, you find thymine pyrophosphate acting there as a coenzyme. So that is on coenzymes and cofactors. Let me see quickly. Somebody has written something here. Say, how do nutritional deficiencies in coenzyme or cofactor precursors impact human health? And that is Sika Augustine. So as I was explaining that cofactors, coenzymes, they act as activators of enzyme biochemical reactions, so that a lack of, say, these vitamins, which are acting as coenzymes, will mean that there will be some malfunction of the enzymes that use these coenzymes as the activators. So you find such deficiencies acting in a way that it renders enzymes that need them as activators malfunctioning. So that is that. So yes, you see you see the deficiency of these either metal ions, the cofactors or coenzymes, manifesting in different ways. Sometimes maybe lack of iron, for instance. You see the same thing you see is that the person is having some form of anemia, but that is because the metal ions are involved, the ferrous ions are involved in the structure of hemoglobin, the black pigment responsible for the transfer of oxygen to the tissues. So you see it as anemia, but you see, if you take it ahead, then you realize it can result into hemolysis, breakdown of red blood cells. And then it also goes ahead and you look at the fact that if hemoglobin is involved in the transfer of oxygen, then it means that there will be impairment of the transport of oxygen to various tissues. So that can begin to result into the death of some tissue cells, some suffocating symptom, which is almost like a loss of breath, sort of not having enough of breath. So on and on and on, complications just with one particular metal. You go on and on and on, and then you see different things, complications that are occurring. Right. And then if you take, for instance, vitamin B9, which is folic acid, for instance. Then we just talk of the fact that this coenzyme is involved in cell proliferation. So it's very much involved in development, so that the lack of it can result into growth retardation if it is in the development of the fetus in pregnancy. Then of course that can even result into some mental retardation, things like that. So it's a complication of different conditions that can occur with the lack of some coenzymes or cofactors, ions, or these coenzymes, which are mainly vitamins. So on a basic level, the impact of these coenzymes you can refer to as a deficiency in vitamins. In general, you can narrow it to specifically what we just did today with vitamin Bs. So deficiency to the vitamin Bs, for instance. Of course, other vitamins also serve as coenzymes, like the vitamin C. They will also serve as some coenzymes. Vitamin D also. So vitamin K, all of these. So when you look at the functions of all of these vitamins, you come to realize when we are referring to the coenzymes, we are mostly just referring to the vitamins. All right, we continue to look at factors affecting enzyme activity. So, Sika Augustine, I hope that is clear. We continue to look at factors affecting enzyme activity. The first factor we're looking at is enzyme concentration. And we see it being depicted here that the rate of reaction of the rate of an enzymatic reaction is directly proportional to enzyme concentration. So what this basically means is that the rate of a chemical reaction will continue to increase linearly when the concentration of an enzyme is increased in that reaction system. And provided that the substrate upon which the enzyme is acting is in excess. So if there is substrate on which the enzyme will be acting, of course, the rate of the reaction will continue to increase linearly in that manner. Of course, if the substrate is finished, there's no substrate any longer available, then of course there will not be any substrate upon which the enzyme will be acting. So that even if you are increasing the concentration of the enzyme, it will not result into increasing the rate of the reaction in any way, because there is no substrate to be acting on. So that is what I meant by provided you have substrate in excess, which means the substrate concentration is more than the concentration of the enzyme. Once you continue to increase the concentration of the enzyme, you'll find that the rate of the reaction will be increasing. Of course, that will be occurring with all other factors held constant, factors like temperature and pH. When they are held constant and the enzyme concentration is being increased, you'll find the rate of the reaction will also be increasing. So that's one factor, enzyme concentration, acting on the activity of enzymes. The second factor is the substrate concentration. So we learned already, the substrate is the compound on which the enzyme is acting. So we see in this case, the rate of the reaction increases up to a point where a steady state is reached. So what I mean by steady state is that you reach a point where there's saturation of the system such that all the enzymes that are present in the reaction mixture are occupied. So that when you continue to increase the substrate, there will no longer be free enzyme molecules that will be acting on the excess substrate. In that case, the rate of the reaction becomes constant. Whatever increase of substrate you do, that will not result into an increase in the rate of the reaction. That saturation point that you reach, that the rate of the reaction becomes constant, we refer to as a steady state. So the substrate concentration, with substrate concentration, you have concentration increase up to a point, and then you reach a saturation, and nothing happens again. Whatever amount of substrate you continue to add, increasing the concentration, it will not result into increase of the rate or reduction of the rate. It will just be a constant rate of the reaction. That will be the maximum rate of the reaction reached. Next is temperature. So we have seen earlier that heat can result into denaturing of proteins. And we see that we are still referring to proteins when we talk about enzymes. We saw that most proteins are enzymes, and just a few of them are some catalytic RNAs. And these RNAs also, of course, they can also break down under high temperature. Now we see that if you take ourselves, our body temperature, it's about 36.6 degrees Celsius, approximately 37 degrees Celsius. That is the optimal temperature for the functioning of our organism. And in fact, most enzymes are optimal at this temperature, say 37 degrees Celsius. Now, at lower temperatures than 37 degrees Celsius, enzymes tend to lose their activity. So the activity of the enzymes gets slower at lower temperatures, that is below this 37 degrees Celsius. So we see the curve showing that as the temperature increases, let's say from 20 degrees, the activity of enzymes or the rate of reaction that the enzymes are catalyzing increases all the way to maximum, which is around that 37 degrees Celsius. And when temperature increases above the 37 degrees Celsius, say somewhere around 40 degrees, you begin to experience a decline in the rate of the reaction all the way. Now, from that, the optimal point down all the way to about 50 degrees, what is happening is that these enzymes, which are proteins, begin to break down, denaturing occurring, and so they start to lose their functionality, so that the rate of the reaction from that point begins to get lower and lower and lower. Now, you see the practical application of this is that, you see, we preserve food with the process of refrigeration. process of refrigeration. And what is the purpose of preserving food? The purpose of preserving food is to keep the food for a longer time, keeping it away from spoilage. And you need to understand that what causes food spoilage is the microorganisms that are present in the food. So microorganisms are all over. They are all over us, in the atmosphere, you prepare food, you put food down. I mean, there will be some microorganisms that will settle in there, or even foods that are canned and you know, packaged in a way. You see, they write some expiry dates on them and all of that. That is because over time, the microorganisms that are present in those food substances will begin to increase in number to the point that the food becomes unwholesome. Now, back to refrigeration. What will cause the food spoilage is that the microorganisms that are present in the food, they feed on the food. And when they feed on the food, they release byproducts or metabolites into the food. Because once a living organism is eating, that living organism will be releasing waste. And it is the waste of the microorganisms, which are toxins, into the food. That is what will cause the food unwholesome for us human beings. When we take in those foods, we can have food poisoning. Now, what refrigeration does is that it lowers the activity of the enzymes of these microorganisms, so that the rate of metabolism of these microorganisms is reduced low to the point that the food can be preserved for a longer time. So it isn't that the microorganisms are not in there, and it isn't that the microorganisms are still not undergoing some metabolic processes. They are still having that, except that the process is now slowed down. So the food can still get spoiled as it is refrigerated. It can still get spoiled. The only thing is that it will take a longer time for the food to get spoiled. So that is what refrigeration does. Refrigeration or freezing will do in terms of food preservation. If you look at, say, conditions of fever, for instance, that somebody is having fever and it's running temperature, the temperature increases going to 40 degrees. That quickly, is attended to by medical practitioners. Quickly, you want to do some sponging. You want to give some prescriptions, all to lower the temperature of that patient. And that is because just increase in temperature alone can result into serious problems, because then you start to have the breakdown of proteins, denaturing of proteins begin to occur. So, and one of such proteins is enzymes, which are involved in biochemical reactions. And once those enzymes begin to break down, as you see, rate of biochemical reactions begin to slow down. And even if you refer to, say, like red blood cells, for instance, you increase in temperature, that will be causing the breakdown of these red blood cells, and eventually will result into some condition of jaundice. You start to see yellow eyes, or some yellow pigmentation of the skin and things like that. And that is because once red blood cells begin to break down that way at a high rate, you start to have a byproduct of the breakdown of hemoglobin. say bilirubin, for instance. So starts having secretion of these things in urine. Urine becomes so yellow, and the breakdown also, result into the eye coloration being yellow, skin coloration being yellow because of the byproducts of the breakdown of the red blood cells. So similar things will occur with a breakdown of enzymes when you have increase in temperature, body temperature, for instance. And once you have the breakdown of these enzymes, obviously the rate of biochemical reactions that are supposed to be regulated by these enzymes will also reduce. Right. Now, pH. pH. So practically, let's say in a buccal cavity, our mouth, in the mouth, pH is hovering around, neutral pH. So the pH of the mouth can be around, say, let's say slightly acidic, like maybe 6.5 thereabout, and then about 7 point something, 7.3, 7.5. So it's hovering around that neutral pH, sometimes slightly acidic, sometimes slightly basic. So it tells you that salivary amylase, enzyme involved in the breakdown of carbohydrates in the mouth, will operate at this pH range. So that's the optimal pH for this enzyme. But let's say we take the stomach. In the stomach, you have, much more acidic pH there, because of course in the gastric juice you find hydrochloric acid is secreted into the gastric juice there, and the pH is much more acidic, somewhere maybe hovering around 2. It can go maybe up to 1.5, come to 3.5, something like that. So there too, it means that the enzymes that act on proteins in the stomach, like inactive pepsinogen getting active by exposure to that acidic medium, becoming pepsin to act on proteins, breaking these peptide bonds in proteins, mean that it is that pH which is optimum for those enzymes involved in digestion in the stomach. So for every enzyme that you take, depending on the enzyme, there too you have some optimum pH. It's not going to be the same for all all enzymes. It depends on the type of enzyme that you are referring to. Of course, we are referring to human beings. Like as I talk about these things, I use ourselves as human beings to do these examples. But of course, there are some microorganisms. There are some bacteria that live in some extreme conditions. So even for temperature, you have some thermophilic bacteria that some you find say deep sea, like if you go onto the FPSO Nkrumah, where they are mining crude oil, down there you find some thermophilic bacteria that can they have optimum temperature for their enzymes, optimum temperature at which they are living somewhere around even the boiling point of water, like 100 degrees Celsius. These are thermophilic bacteria. They live at such very high temperatures. Same way you can have some microorganisms, again, some bacteria that can live at some different acidic conditions, very low, very, very low acidic conditions, like acidic conditions of 2, pH of 2, like that is their optimum at which they live. So you can have these different conditions that are affecting the rate of the activity of enzymes. All right, so I see, and then there's a question from Owusu Mary, and she says, please, since rate decreases at both lower and higher temperatures, is there any means by which the rate may increase due to temperature, or can rate of reaction increase with temperature without it decelerating? So I think that is just, I didn't even see this before I started talking about some thermophilic bacteria that can live at very high temperatures. It will mean that at those high temperatures, that will be the optimum for their enzymes to be acting. Now, particularly when we are referring to enzymatic reactions, it means the catalysts for these reactions are the enzymes, and these are proteins, and these proteins will have their functional structures at some optimum temperature, so that when that temperature increases, the protein breaks down and loses its functionality. So if you increase temperature, obviously you break down these enzymes. So if you are running enzymatic reactions in the laboratory, you want to stay within the limit of temperature, so you don't break down the enzymes and lose everything. Like the whole reaction may shut, then nothing will happen. You have to start that experiment all over again. It's different from when you are running non-enzymatic reactions in the lab. So enzymes are not involved. It is actually, let's say, increase of temperature is a form of activation of reaction molecules, or let's say you give some, if I cause a particular reaction to give you some complex which is being formed. Apart from that, if you are increasing temperature as a form of activation of molecules, like you're trying to catalyze a reaction by using temperature to increase it, then it shouldn't be an enzymatic reaction. But even in that sense, you have to pay attention to things like, for instance, the melting point of the various reactants that are involved. Because if you increase temperature above the melting point of, say, 1 or 2 of the reactants, obviously you'll be breaking down, you'll be degrading the reactants. So the reaction will not proceed, because you will just break down the reactants by increasing the temperature above its melting point. So the reactant will just get degraded, and then you don't have the reaction occurring. Somebody says my network is not stable. I'm monitoring from here. I think my network is stable. Perhaps it's yours. If my network is unstable, it will show on my screen, and even I see it. From the beginning, it wasn't stable. Sometimes it drops a bit. When it drops, I keep quiet for some time. Then when it comes, I continue. But now it's stable. All right. So I hope, Owusu Mary, you are okay with the question on temperatures. Okay, so we can move on and take a look at mechanisms of enzyme action. mechanisms of enzyme action. Now, the first understanding of enzyme action, postulated was that of the lock and key model, which assumes that the enzyme and a substrate behave like the lock and the key, that such that the active center of an enzyme is shaped such that it fits perfectly with a particular substrate. So a particular substrate that is having a certain structure is fit for the active center of an enzyme. So we see it being depicted here, the key being the enzyme and then the substrate there, and the locking well fit, so that we see it also being shown here as so this is an enzyme molecule which is having the active center structured in such a way that this enzyme is supposed to catalyze the reaction of two reactants. So two substrates are to bind in there. So we see one of the substrates is this this black round circle that or this black sphere that can bind to some portion of the active center of the enzyme. And then we also see some grooved square that can also fit at another portion of that active center. So these two portions are part of the active center. One of them for this black sphere and one for this grooved square. And the two of them bind to form enzyme substrate complex, so that the enzyme substrate complex, which is what we'll refer to as a transition state, then you have the two substrates binding to form a product, which then dissociates away from the enzyme. So that's a model, the first model that was used for the studies of enzymes, referring to a particular portion of enzymes, which we refer to as the active site. We talked about the active site when we were looking at the terminologies in our first lecture on enzymes. But there's a problem with this model. And so if we take, say, a rod, let's say some rod which is being referred to as the metal stick as a substrate, and that metal stick is to be broken. So let's say that's the reaction we want is to have that metal stick broken. In a transition state, the metal stick is bent, and then from the transition state, you have the metal broken into two. So you have this broken stick as the products. Now, with this analogy, if you think of an enzyme being complementary to a substrate, like absolutely complementary, fitting, just like how we saw the lock and key fitting together, this is what you see. So if you take the enzyme as a magnet, which fits perfectly to the substrate to form an enzyme substrate complex, this fits so perfectly that there will not be any need for a product to be formed. Because what you see here is a stable complex, very stable, so stable that there will not be any reason for which this enzyme substrate complex will want to form any product to change into any other form, because there is that stability. The energy level at which it reaches here, the enzyme substrate complex, is so stable that there will not be any form of a reaction occurring in that in that sense. And so from here, that lock and key model had been modified to say that, look, the enzyme substrate complex which is formed is actually not that fit, that it is so stable, but it is rather the transition state that fits in there, so that you see in the beginning, you have the enzyme being shown as a magnet, not so fit, but you see the metal rod in there having an enzyme substrate complex, will then move to a transition state that bends that metal stick. And because it is bent, obviously, it can easily be broken into the products, and that metal stick then dissociates away from the enzymes. You have your enzyme, and then you have your product. So that was a modernization of the lock and key model, and that led to the current model which is used in enzymatic studies, which is referred to as the induced fit model. And that induced fit model refers to the fact that a substrate actually induces an enzyme to have some conformational changes in its active site, so that the active site now fits to the substrate. So what you see here is an active site depicted as having some round shape, which does not fit the square shape of the substrate. But once the substrate moves to bind to the enzyme in that active site, conformational changes occur so that the active site of the enzyme is now changed to fit the substrate. And so you now have the enzyme substrate complex fit for the reaction to proceed to give us products that dissociate away from the enzyme in the end. So this is like the coming from the lock and key model. We have the induced fit model, as a modernization of that lock and key model, to say that what is actually happening is that a substrate induces the active center of an enzyme to fit, or a substrate induces conformational changes in enzymes, so that the enzyme now fits to the substrate to act. Of course, it's not that any substrate at all will be causing conformational changes in an enzyme, but there is that specificity. We refer to specificity. So a substrate of a particular enzyme can induce such conformational change in that enzyme for the enzyme to fit, and then you can have the catalysis of the reaction to occur. Then we also have acid-base reactions that occur in the active center of enzymes. So starting from the beginning, we looked at 20 amino acids which are found in proteins. And out of these 20 amino acids, we classified some of them as having acidic side chains, and we classify some of them as having basic side chains. Now, enzymatic studies, have revealed that 5 of these amino acids are dominant in the active centers of enzymes, and these are histidine, cysteine, aspartic acid, arginine, and glutamic acid. These 5 are found to dominate the active center of enzymes. And in their functions in that active site, you realize the reactions that take place in the activity of the enzymes involve, acid-base reactions, which we refer to as acid-base catalysis. So we see an example of an enzyme, papain, or you can say papain, which is involved in the breakdown of peptide bonds. And actually, papain is used, practically in meat tenderizers. So in meat processing industry, this is an enzyme which is used to soften meat. So we have it in like meat tenderizers. Now we see at the active center of papain, which is shown here, we have a cysteine residue. That's an amino acid. And if you take a protein, the protein is made up of amino acids, and you can actually, if you look at the primary structure of the protein, you say you have sequences of amino acids. So you can say this amino acid comes first, this one comes second, this one comes third, all the way. That way, when you do that counting, when you are reading the biochemistry books and you see things like this, see 25 is written, it's only showing that this cysteine residue is at the 25th position in that primary structure in the sequence of amino acids. The same way, histidine is at 159th position. But you see, even though they are at 25th and 159th position far apart, you see that the protein folds in such a way that cysteine and histidine have come close to each other, forming the active center there. Now, in this case, we see the cysteine molecule that is having sulfur in it, and you see histidine is having a ring. We refer to this ring as an imidazole ring. And you see that the nitrogen of this ring is having some hydrogen bonding on the amine group of some amino acid there, which is bonding to the carboxylic acid group of another amino acid. So the peptide bond is formed between the carbon and the nitrogen there. It is that peptide bond that is to be weakened and broken. So we see there are some lone pairs of electrons on sulfur, which is having some nucleophilic attack on the carbon of that carbonyl group, which is in that peptide bond. So in effect, you have some sort of stretching of the peptide bond, weakening it, and it can eventually be broken. That is an acid-base catalysis because you see the nitrogen here that is having some hydrogen bonding of the hydrogen on that amine group. In a way, it is a form of accepting a proton from that amine group. So it's acting as a base to accept that hydrogen or the proton from there in that case. And eventually the neutrophilic attack of this lone pair of electrons on the carbonyl group of that peptide bond can weaken the peptide bond and break it. Now, basically, what happens in the active center of enzymes with acid-base catalysis is, say, hydrogen ions or protons are donated from one amino acid to the other. It's some form of activation that occurs inside the enzyme for the enzymatic reaction to occur. All right, I see somebody says, please, sir, is there an inorganic enzyme in nature? And how does it differ from organic enzymes in function? So I don't mean I don't really get what you mean by an inorganic enzyme in nature, because in living systems, enzymes are mostly proteins. Proteins are organic substances. And apart from that, we also saw that you can have some nucleic acids that are catalytic. They can also, you know, increase the rate or say catalyze reactions. Mostly it is in the breakdown of nucleic acids themselves that they are involved. Okay, you said could they be made in the lab? Okay, so you're thinking of an enzyme that can be synthesized in the lab. So that would be like some peptide-like molecule. Of course, that can be done. That is what is actually done in let's say in pharmaceutical industries, that an enzyme is known to be involved in some particular reaction, and the that the enzyme to be like you want the enzyme to be produced in large amounts, and perhaps you want to have some tweaking of the enzyme to perform some specific function. You do some synthesis to have some derivative of some amino acid, for instance, to carry out some specific action. But that is sort of too expensive, too complicated to be doing. So what pharmaceutical industries will actually do is that they will rather employ microorganisms. So if you do biotechnology, for instance, you see that you have these bioreactors that you only need to provide optimum conditions for microorganisms to produce a particular enzyme that you need in large amounts. You purify that, and then you use it in your pharmaceutical product, for instance. Then that is it. Instead of wanting to be doing synthesis, that would be too expensive, and even because you now have to be running reactions with some organic compounds, it's just too demanding to be doing such a thing, and the product in the end is going to be too expensive. So you rather employ the microorganisms as a micro factory, that if you just provide optimum temperature in some industrial reactors, they begin to produce these enzymes in large amounts, and then you just extract them, purify the protein as the enzymes, and make use of it. And in that sense, those enzymes they will not differ from the enzymes that are produced in the organism itself, because of course it is the organism which is producing the enzyme, and you are just collecting it. You only provide condition for the enzymes to be produced in large amounts. I hope it's clear. Okay, so that is about acid-base catalysis, which actually occupies, like, now most of the enzymatic reactions studied lots of times. It's about this acid-base chemistry. Somebody says, Evans says, please, the acid-base catalyst, do they accept and donate protons? Yes, exactly. So you have an amino acid residue which accepts, and you have an amino acid that donates. So you have a proton donor and a proton acceptor in the active site. So exactly what it is. You have a proton donor and you have a proton acceptor in there. Okay. These studies of enzymatic reactions these days, if you were doing computational chemistry, you actually see these things, just on a computer screen. That there are softwares that you use to model the molecules, and you see the interaction of the molecules, how the reactions are occurring at different energy levels. Is actually one of the ways that, let's say, pharmaceutical companies, they run some preliminary computational modeling to eliminate some molecules that are of interest, that in the end, let's say the computational modeling shows that they do not have prospects in the studies that they are doing. So they are eliminated out of the process and then left with molecules that are more prospective, which they will now, uh, continue to study with wet chemistry, that is now in the lab in test tubes to now test them. So it reduces the amount of work with trial and errors, that you are thinking of different molecules that you can use to microphone went off completely. All right. Can you hear me now? I hope you can hear me now.
Yes, sir.
Okay, great. So that's good. The lights in my area went off just a while ago, and so I had to quickly put on some backup internet. So that's why I went off a bit. And by the time I came, I think the microphone automatically went off. So when I was talking, you couldn't hear me. But we are back now, so we can continue. Just a moment. Let me see. Okay, so then I think the previous file, what I was saying, the previous slide, what I was saying before, you couldn't hear. I was saying that, you know, I was talking about computational chemistry, which is used to study some of these enzymatic reactions, looking at, you know, modeling of the molecules and seeing the sort of interactions they are having at different energy levels, which is used to eliminate some candidate molecules in trial and error so that it reduces the number of molecules that are tested as some potential drugs. So it is only those that are seen to be prospective that are then, you know, studied, you know, practically now in wet chemistry, in test tubes now to actually continue in the process of, you know, the drug discovery process. So those of you who will be interested in, you know, biochemistry in the future, so one of the fields you can go into is computational chemistry. All right, so we just take a look at the activity and like practically the activity of another enzyme, which we calling lysozyme. Lysozyme is involved in the breakdown of the cell walls of bacteria. So in some antibiotics or some bactericidal agents, lysozymes, you know, break down the glycosidic bonds that are found between the monomers that come together to form the cell wall of bacteria. Hey, somebody says you can't see my screen. Is it that? But I'm still sharing, so I hope you can see now. Awinam says she can't see. Elon also says she can't see. But I'm still sharing the screen, so. I'm sharing now, so can you see it? Maybe it takes a little time to come. Okay, great. Nicola says you can see, but somebody also says, I think some of you, your network is not, it's also not stable. That is why it keeps. Okay, great. But whatever it is, this video is being recorded. So after the lecture, I will share the link. You can watch it. You can download it and watch it many times. So that some of these breakages, you can still deal with it. So I was saying the bacterial walls are made up of monomers, like two monomers, which is N-acetylmuramic acid and N-acetyl glucosamine. So we see the bond. So these are like these are glucose molecules, derivatives of glucose molecules. So you have muramic acid, and then you have N-acetyl muramic acid, and you have N-acetyl glucosamine, and between them you are having a glycosidic bond, and it is shown much clearly, magnified in this circle, that you are having N-acetyl muramic acid on the left-hand side, and then you have N-acetyl glucosamine on the right-hand side, and between them you have a glycosidic bond, And it is this glycosidic bond that gets broken by lysozyme. So it's a hydrolytic reaction, and that now results into a single N-acetyl moramic acid and N-acetyl glucosamine on the right-hand side. So the bond is broken between them. Now, this structure, what you see on your screen, the structure is actually the structure of the cell wall of bacteria, which is made up of these alternating molecules of moramic acid and acetylglucosamine, with the bond between them being a glycosidic bond. So this way you have lysozyme, which is a bactericidal agent, can break down the bacterial walls of bacteria, and in essence, sort of, destroy that bacteria. So that is another application, a practical application of enzymes in, say, antibiotics or bactericidal agents. All right. Another thing we can look at is the slowing down or the blocking of enzymes, prevention of enzymatic reactions, which we refer to as enzyme inhibition. And this can be done by molecules which we refer to as inhibitors. So molecule that inhibits enzymes, either it slows down the enzymatic reaction or stopping the enzymatic reactions from occurring. These are inhibitors, and these inhibitors can also be used as some pharmaceutical agents. So we have examples like painkillers such as paracetamol and aspirin. They inhibit, enzymes, some oxidases that are involved in the synthesis of some signaling molecules, prostaglandins, which are some derivatives of fatty acids. And these prostaglandins are involved in processing of pain, like they are involved in inflammatory reactions. So they will, sort of either cause some dilation of blood vessels or constriction of blood vessels. And so they will be involved in causing swelling and pain at some places where you have some inflammation. So that if the enzyme which is in that all these oxidases which are involved in the synthesis of these prostaglandins, when they are inhibited, it will reduce the amount of prostaglandins which will be synthesized. In that sense, then the individual taking in the paracetamol or aspirin will have some pain relief. So we can look at enzyme inhibition in different ways. We have what we refer to as competitive inhibition. Competitive inhibition occurs. So we're looking at competitive inhibition. Competitive inhibition occurs by, say, the substrate and the inhibitor competing for the active site of the enzyme. So in competitive inhibition, what you see on the right-hand side is that the inhibitor, which is depicted I, competes with the substrate for the active center of the enzyme. So if you have the inhibitor binding to that active site, it means it has blocked the active site, and thus the substrate cannot bind onto it. And once the substrate cannot bind onto it, it means that reaction has been prevented. the reaction of an enzyme acting on a substrate, giving you enzyme substrate complex, which is the transition state before you have a product formed, which dissociates away from the enzyme. That reaction is prevented from occurring because the inhibitor competes for that active site, binds to it, and prevents the substrate from binding to the active site. Then we also have what we refer to as uncompetitive inhibition. Uncompetitive inhibition. So in uncompetitive inhibition, apart from the active site where the substrate binds, there is also another site, usually we refer to as an allosteric site, to which the inhibitor binds. So even though you find the enzyme is binding to the substrate to give you an enzyme substrate complex, the inhibitor comes to bind to that allosteric site, which then prevents the reaction from proceeding to products. So somebody said inhibitors, they bind to amino acid. Please, I want to know the type of bond binding them. Okay, so we'll come to the type of bond. As we go ahead, we'll see the type of bond that can occur between the inhibitor and the enzyme. Mainly two types of bonds can be there, or some weak bonds, some. So we'll see. We'll see them as we go ahead. All right. I noticed that when I move to a next slide, it takes some time for the slide to move on your screens. So now it has come. So I'm monitoring it with another device so I can see it clearly. Now, we have also what we refer to as mixed inhibition. Mixed inhibition. So in mixed inhibition, you see there's an enzyme that can bind to the active center. The substrate can bind to the active center there. So if you have an enzyme substrate complex before the inhibitor comes to bind to that enzyme substrate complex. So you now have an enzyme substrate inhibitor complex, which cannot proceed to products. You can also have the inhibitor binding first. So you now have an enzyme inhibitor complex before the substrate comes to bind to the active center. All of these will end up with the enzyme substrate inhibitor complex, which will not proceed to products. These we refer to as mixed inhibition. So if you take a look at it again and you realize if you're looking at the reaction first horizontally, that the substrate binds first to the enzyme to give you an enzyme substrate complex before the inhibitor binds to it to give you enzyme substrate inhibitor complex, which does not proceed to product. Or if you look at the reaction vertically first, where you see the enzyme binding to the inhibitor binds to the enzyme first. So inactivating the enzyme in the first place, even before the substrate comes to bind. So when the substrate binds, it forms the enzyme substrate inhibitor complex. These two together will be occurring at the same time, and this we refer to as mixed inhibition. Now to the bond, the question that Evans asked. Evans asked a question about the type of bond which is formed between the inhibitors and the enzymes. So we can have covalent bonds that are binding the enzyme and the inhibitor. In this instance, we refer to such inhibition as irreversible inhibition when covalent bond forms. Any other bond which is non-covalent usually maybe some van der Waals forces, could be some weak hydrogen bonds, things like that. Those bonds are involved in what we refer to as reversible inhibition. That is reversible inhibition. So an example of reversible inhibition we saw earlier was that of succinylcholine right from the beginning. Succinylcholine, which was binding to the receptors of the muscle cells, such that, so in a way, it was inhibiting the acetylcholine esterase from, it blocks the receptors from now, the binding of acetylcholine esterase for the muscle to contract again. But the succinylcholine does not stay there forever. Remember, those proteins which are the receptors are also some proteins that the succinylcholine is binding to, except that in this case, succinylcholine is not binding directly to the enzyme, the acetylcholine esterase, but it's rather blocking which the choline esterase should be acting. But it doesn't stay there, forever. It doesn't stay there permanently because it's not covalent bonding. That is a non-covalent association which is formed in that instance. That is why after the surgery, there is a reversal of that effect, so that muscle contraction and relaxation can resume. But you have irreversible inhibition, in which you have covalent bonds forming. And when covalent bonds form, it means that becomes permanent, and the enzyme is rendered completely non-functional in the end. That obviously will be the case of the destruction of some pathogenic bacteria, for instance. Right. So we look at an example. A practical example of such is that of penicillin, an antibiotic, and we talk about the fact that bacteria is having its cell walls being made up of peptidoglycan, and peptidoglycan is what you see, what the components of it being N-acetylmuramic acid and N-acetyl glucosamine, which are having the glycosidic bonds between them. Now, in the formation of peptidoglycan, that structure that you see, that we saw earlier, that lysozyme was breaking the glycosidic bond between. In the formation of it, you have the enzyme glycopeptide transpeptidase that is involved in the formation of that bacterial cell wall. And we are here. You see the OH you see here is to depict a serine residue, the amino acid serine. On the side chain there is CH2OH. So the OH is just depicting that end of that serine residue. Now, what penicillin does, so this is the structure of penicillin. Penicillin comes to bind to that serine residue, and that serine residue is in the active center of the glycopeptide transpeptidase. And penicillin comes to bind to that active center of this enzyme, forming this inactive penicillyl enzyme complex, so rendering the enzyme non-functional. And this bond is a covalent bond, and so that inhibition is a permanent inhibition that results into the destruction of the bacteria in that sense. We refer to these irreversible inhibitors, suicide inactivators, because they eventually this is going to result into the death of the bacteria. After you have the lysis or the breakdown of the bacteria walls, eventually that results into the death of the bacteria. All right, so this is where we end with enzymes. The next time we meet, we're going to take another protein, which is hemoglobin, and we look at the structure and the functions of hemoglobin, just like we have done with enzymes. So now I can. So somebody says, "Sir, please, can it be called a zymogen?" Well, zymogens are the same as non-active proteins, sort of like how we have pepsinogen that is activated into its active form, which is pepsin. Those are zymogens. So this is not a zymogen. It's not a zymogen. Okay. If there are any questions or anything, so I'm looking at the chat. So if there is any question, I can respond to it.